Hello everyone, my name is Ian McLaughlin and I have a PhD in neuroscience, and today I'd like to talk about a topic that I've discussed a fair bit in the past, a substance that's undergone a bit of a renaissance over the past few years, cannabis. So cannabis is a massive topic, right? And its pharmacology is extremely complicated, due in part to this wave of state-level legalization that's happened over the past, kind of a long time, actually. Despite that sort of wave of legalization, the status of cannabis is still quite illegal at the federal level. And there's been this sort of sustained and growing interest in understanding the basic neurobiology of how it is that this complicated plant affects the human nervous system. And so to give you a sense of where the country is, and once again, I apologize for being rather United States-centric in the beginning here, but I think this is kind of an unfortunate case of U.S. policy driving international policy, but to give you a sense of where the country is, you're obviously looking at a map of the United States, and you're seeing 4 different colors that indicate 4 different legalization statuses. Totally illegal, gray. Legalized for medical use, that sort of aquamarine-ish color. Decriminalized, that sort of dark blue-greenish color. And then legal for recreational use, green, bright green. So I happen to know that this map isn't entirely up to date because I know that New Jersey would be that bright green color as of a few months ago. But I think this pretty much conveys the point, right, that the number of states in which cannabis remains entirely illegal is in the very, very small minority at this point. Only 6 states. The rest have some form of legal status assigned to cannabis. Now, of course, that doesn't speak to the state of affairs at the federal level, right? Which is an entirely different can of worms, or weeds, I guess. But I think you get the point, right? The vast majority of states have found a reason to provide for some legal consumption of this substance, either exclusively for medical reasons or irrespective of whether it's, for medical or just purely recreational reasons. There are a few holdouts, obviously, but I think we can see the way the wind's blowing here. But if we look to the rest of the world, we get a bit of a different picture, with all of that bright red indicating countries where cannabis is both illegal and that illegality is enforced. Pink is where it's technically illegal, but that illegality isn't really meaningfully enforced. It's on the books, but nobody really cares. Orange countries are where cannabis is technically illegal but decriminalized, so sort of functionally legalized. And then in blue, we have countries and some states where it's straight up legal. And then, of course, in gray, we have countries where exclusively penguins smoke pot. But, okay, so I'm unsure how up-to-date this world map is, but I imagine it's fairly accurately representing the state of affairs globally. So, a lot of progress to be made internationally regarding the use of this substance. Okay, so as I mentioned, the pharmacology of cannabis and cannabinoids, right, THC, CBD, is pretty complicated. In short, cannabis is a plant, obviously, but it produces a huge variety of molecules, some of which have interesting pharmacological properties, right? The most well-known molecules produced by cannabis are likely familiar to anyone who might be listening to this, who might have clicked on this. THC, or tetrahydrocannabinol, and CBD, cannabidiol. But there are tons of cannabinoids that have been identified in this plant. And honestly, I've seen different studies claim different numbers of distinct cannabinoid molecules within the plant, and I'll link at least one of them from just a few years ago, I think 2017, that claims that over 100 have been identified. But for our purposes, the point is that there are many more cannabinoids produced by this plant than just THC and CBD. But beyond solely the cannabinoids like THC and CBD, the plant produces a whole variety of additional molecules with interesting properties. And it's those sort of fatty, sticky molecules on which this study focuses. This is a study out of the University of Arizona, and it's really a behavioral and electrophysiological study, right, where scientists try to get a sense of how it is that various molecules affect humans by seeing how they affect certain animals, and in this case, our cherished biomedical ally, the mouse. And the group did at least one of the same behavioral tests that I used to do when I was in the lab, and also happened to use the same software that I used while I was doing my research. But anyways, I know that electrophysiology is probably one of those words that isn't particularly meaningful to a lot of people. It's just when neuroscientists use tools to get a sense of what very specific parts of our brain, specific brain cells, or even specific parts of brain cells, to get a sense of what those very specific parts of our brain are doing. Okay, and so we can get a sense of how, when one out of our approximately 86 billion neurons becomes particularly chatty, right, active, causing a bunch of other brain cells, neurons, to either become super active or completely shut down and become silent. It's sort of like if you're standing outside of a room, right, in which people are having a conversation, and you really want to know what they're saying, but you just can't hear a single thing, right, because the door is closed. And then all of a sudden you stick a microphone on a long pole under the door, and then all of a sudden you hear every word, right? That's kind of like what electrophysiology is about. Kind of, sort of. Okay, so this is a group who's getting a sense of how it is that specific molecules change behavior, okay? And they're also getting a sense of how the molecules change activity in the brain, okay? Pretty cool. And it's basically what the next paper on which I'm an author did, but different drugs and different brain regions. But okay, so once again, what makes this study interesting isn't that they're focusing on cannabis, which is a substance that's been receiving some of the greatest rise in attention in biomedical science, right? And more importantly, enjoying this wave of state level legalization. What makes this study interesting is that they're focusing on the terpenes that the plant produces. Okay, so what on earth are terpenes? I just got comfortable with cannabinoids. Now I have to learn about terpenes. So these are the things that the plant produces that makes cannabis have that immediately recognizable smell. Okay, and to people who are sort of aficionados of the substance, these are the molecules that distinguish varieties of cannabis or strains of cannabis that have unique olfactory or odorant characteristics. Right, they're what make cannabis smell citrusy or piney or berryish or minty, skunky. And you could, there's almost an infinite number of words that you could assign, and you can see here they're breaking it down into spicy, sweet, sour, and earthy. But depending upon specifically which combination of terpenes are present, you'll get differences in smells, right? Sort of like how a Honeycrisp apple tastes both similar as well as quite different relative to like a Red Delicious apple, which, by the way, I'm continuously baffled when I see somebody actually still buy a Red Delicious apple instead of like a nice Honeycrisp or Cosmic Crisp or Sweet Tango, which is my favorite. But anyway, so it's sort of like that, right? And and just to be clear, terpenes are found all over the place, right? They are largely produced in plants. More on that later. But there are even terpenes that are produced by animals, like squalene, for example. So squalene is one of those words that's just like aesthetically unfortunate in my opinion, but it's in a ton of sort of trendy skincare products, right? So squalene is a terpene, and while it's found in plants, as you can see, I think this one's plant derived, it was originally derived from shark liver oil, and so it's also very important in the synthesis of plant sterols, right? Like cholesterol, as well as the steroids that our own bodies produce, and so it's a pretty important molecule, important enough that both plants and animals produce it. Squalene, a terpene. Anyway, so these molecules, the terpenes, which are the again the sticky, fatty kind of substances, are produced by the same parts of the plant that produce molecules like THC and CBD, trichomes. Okay, so that term trichome is actually derived from a Greek term referring to hair, and you can sort of see why in these pictures. So there's sort of like little protuberances that look almost like fur on a plant, but they contain a whole variety of molecules that, at least in some plants, like cannabis, for example, have some interesting interactions with the human body and the human nervous system, right? And by the way, they're sometimes called like resin glands, or just resin by sort of devotees, by the community of folks who consume cannabis, but same things. Okay. And for my fellow plant nerds, trichomes come in many shapes and sizes. Right, so it wasn't, not that many shapes and sizes, but everything from algae to lichen to plants have trichomes, and only some of which are glandular. Okay, which is what's going on in cannabis. Right, the whole concept is that in plants with glandular trichomes, there are interesting molecules secreted within them. Okay, molecules that are byproduct of the plant's growth: THC, CBD, terpenes. Okay, so So these are the molecules on which this group is focusing, terpenes, right? Of which there are many, by the way, and which determine the unique odors of various strains of cannabis, right? Which are produced in these tiny little hairy thingies on the buds of the cannabis plant, mostly on the buds of the cannabis plant. And by the way, you can think of buds of cannabis plants sort of like flowers, the equivalent of like cannabis flowers. And just to be clear, right, while cannabis is producing terpenes in these trichomes, those molecules aren't necessarily unique to cannabis. You can find those molecules in other plants as well. Okay, so for example, the most commonly or the most common terpene in cannabis extracts, though it's technically a terpenoid because it contains oxygen in the molecule, but it's still in the same category. Anyways, the most common terpenoid in cannabis extracts is present in a ton of foods that you probably eat: basil, oregano, black pepper, rosemary, one of my favorites, and a bunch of other plants contain this molecule, Beta-Carophylene. Okay, and this molecule, which once again is found in all those herbs that are probably in your spice cabinet or whatever, they're used in basically any interesting meal you've ever had. This molecule is a cannabinoid receptor agonist. Meaning that it activates some of the same receptors that the ingredients, the active ingredients of cannabis activate, right? THC, CBD. So these aren't molecules that are only found in cannabis, and they have some interesting pharmacological properties. Okay, there have been some interesting studies that suggest that different terpenes may have interesting biological effects. Things like altering the way animals react to caffeine. And I'll include a link to a great review of these from a few years ago below. It's pretty extensive, but what's cool about it is that it also goes into a bunch of cannabinoids that I suspect are unfamiliar to most people. You just haven't heard of them because not a lot of people study them, and it's also freely available, so you can just snag it. You don't have to subscribe or anything. Okay, so let's finally get into the study for today. Enough background. So here's the main question that this group is asking. Is there an entourage effect? Meaning, does the combination of the molecules that we know are psychoactive, THC, CBD, and so on, does the combination of those molecules with the molecules that give cannabis strains their unique smells and flavors, does that combination actually change the effects in the brain? And when I say effects, the group explored a whole bunch of different effects of cannabis, right? But the main finding, these terpenes, the specific terpenes that the group really interrogated, right? They didn't study all of the terpenes in a strain. They studied a handful, but the ones on which they focused, they actually mimic the effects of THC itself. So, the group honed in on a specific handful of terpenes and terpenoids, which remember, they're just terpenes that have some oxygen in their molecules. Not really important. They're both just fatty, sticky molecules produced in these trichomes. Some of the ones that I find fascinating are linalool, which smells kind of like lavender, or like a citrusy lavender, evidently, as well as beta-pinene, which smells like birch. Now, of course, it smells like pine, and it's a very unmistakable smell, and lovely in my opinion. They scrutinized a couple of other ones. They explored alpha-humulene, which evidently smells sort of like hops, like those hops that are used to make beer, which is a pretty similar plant, by the way, the hops plant, compared to the cannabis plant. But yeah, so evidently the molecules smell sort of like hops, woodsy, earthy kind of smell going on. And then they also looked at geraniol, which evidently smells a bit like roses, but it's also present in citronella oil. Pretty neat, right? And by the way, these molecules tend to be pretty simple molecularly. And so just to give you a sense of what one of the molecules in which we're going to explore or focus a little bit later, geraniol, there we go. Pretty simple molecule, right? I mean, it's not a whole, I guess, and that I suspect would be considered a terpenoid due to that alcohol group at the end there, that hydroxyl group at the end there. Anyways, yeah, simple molecule, right? If you compare that to like the molecules of THC and CBD, which we'll check out in just a sec, this is a very simple molecule, but it has this powerful scent. Pretty cool, huh? Okay, so they honed in on these particular terpenes that I just listed because they're found at particularly high concentrations in cannabis, though some of them are at tend to be at higher concentrations than others, right? Some of them there's just more of one of them than another. But they honed in on those because they're among the more common ones. And there's some sort of complicated pharmacology going on in this study, right? They were working with a cannabinoid that's really structurally quite different, like in terms of its molecular structure, compared to the more sort of standard cannabinoids like THC and CBD. So they were working with something called Win 55-212-2, but we'll just call it Win 55, which is a cannabinoid that's been studied for a long time, not produced by the cannabis plant, by the way. Okay. So, and I know looking at, these types of chemical structures isn't particularly informative. There's a bunch of hexagons and pentagons and stuff with some letters and lines and stuff, right? But let's compare it just qualitatively to THC. Okay. Ooh, it's a little dark. Yeah, you can sort of see it. Okay. So, I know, it's just another bunch of hexagons and lines and stuff. But you don't have to have a PhD in organic chemistry to get a sense that these are pretty structurally different shapes, right? Different numbers of hexagons and pentagons, different letters within that pattern of shapes, right? And THC has this big old long tail, right? While Win55 has a kind of big old long hexagon hanging off of its tail. These are different molecules, right? But both of the molecules bind cannabinoid receptors. And so they were working, they were trying to get a sense of whether terpenes, right, both on their own, as well as in combination with a molecule that isn't THC, right, or CBD. They're trying to get a sense of whether or not these terpenes have their own effects, or if they alter the effects of what happens when our cannabinoid receptors, the receptors to which THC binds, if they alter the effects when those receptors are stimulated, when cannabinoid receptors are being stimulated by a cannabinoid. So terpenes alone, do they have their own effects? And then do they influence the effects of cannabinoid receptor stimulation? Okay, so before we dive directly into what they found, we have to talk a bit about, just a little bit about some terminology that's kind of central to how these scientists interrogated the endocannabinoid signaling system. The entire system that's THC and CBD influenced, by the way, but this system is extremely broad and very complicated and influences a ton of, aspects of our consciousness and our experience, far more than I suspect a lot of folks who have experience with cannabis might even suspect. And so all this to say, we have to talk about the tetrad model. Okay, so this is just a word to describe 4 sort of categories of effects that molecules that interact with cannabinoid receptors induce in animals. Okay, so we have hypolocomotion, which means that animals, including humans, will tend to just sort of slow down and stop moving around so much. We have hypothermia, which means that body temperatures drop a bit. Analgesia, which means our sensitivity to pain tends to decrease, and then catalepsy, which when it comes to this specific application tends to just mean like a tendency to just not be driven to move, even if an animal's in a sort of uncomfortable and awkward position. That's the tetrad, okay? Animals tend to move around less while they're moving. We tend to not really want to move very much at all. We tend to have lower body temperatures, and we tend to have reduced sensitivity to pain. Okay. So here's where this group begins. Here's our tetrad model. I know this is kind of a wacky figure. It's called a radar chart, by the way, and I honestly kind of love it, but I know it's a little bit inscrutable. But so basically, you have the central diamond as 0, no effect whatsoever. The farther you move from the center, the stronger the effect towards one of those cannabinoid-related effects in the tetrad, right? The different colors are representing how significantly each of those 4 terpenes, which you can see listed up top here, how these terpenes how much they induced each of these 4 effects in the tetrad. And so, you can see, for example, that pinene, which is that blue kind of kite-shaped thing, it induced a pretty significant change in how mobile animals were, right? While it didn't really have much of an effect on the other 3 effects of the tetrad, right? But then, if you look at geraniol, that terpene that smells sort of like roses, had a pretty cataleptic effect, making animals just sort of like linger in a body position that's just kind of awkward, like a position that most animals would pretty rapidly move away from. And then, keep in mind that the molecule Win55, right, that molecule that stimulates the same receptors as THC does, but isn't found in cannabis at all, they're comparing changes in these behaviors right? Or yeah, in these behaviors to what happens in animals when they're given Win55. Okay, so in other words, what happens when the same cannabinoid receptors that THC stimulates are activated, but using a molecule that isn't even found in cannabis, right? So it's not an interaction specifically with the THC molecule itself. It's an interaction with the activation of the receptors that THC stimulates, and they're using Win55 to achieve that. Okay, so what we're seeing is that these 4 terpenes, the 4 different colors, polygons here, they seem to influence the sort of classic effects of cannabinoids, but in different ways, right? It's not like all of these polygons are like, completely congruent. They're not like laying direct. You don't just have one color, right? You get different sort of distributions of effects. So what does that mean? So that means that if an animal were to consume or smoke or vaporize or whatever, a strain of or an extract of cannabis that happened to have quite a bit more geraniol, right, that rose-like scent, which to be clear is also used in combinations with other terpenes to achieve a whole bunch of fruity flavors, right, like grapefruit, apple, blueberry, geraniol. Okay, so strains that have high concentrations of geraniol may very well be more likely to cause somebody to just stay stuck to their couch, or couch lock, in other words, right? But if an animal were to consume a strain that happens to be high in alpha-humulene, which is that terpene that has that sort of hopsy, woodsy kind of characteristic, earthy kind of smell, that's also found in basil and rosemary, according to these data, that animal, if they consumed a strain that was high in alpha-humulene, wouldn't seem particularly immobile at all, right? Because you can tell by that green trace, right? They're moving. So that's what this radar chart is showing, okay? And keep in mind, this is in mice, right? And that's always worth remembering that, while our nervous systems are remarkably similar, frankly, there are obviously substantial differences as well between the mouse, central nervous system and the human central nervous system, and nervous system in general. So, these data shouldn't be interpreted as indicating that certain strains, that happen to be high in one of these terpenes will most definitely have these same effects in a human. So, a strain that's high in, pinene or alpha-humulene won't couch lock you so much, whereas, ones that are high in geraniol will, right? We can't necessarily make that go that far in our interpretation. But what these data show is that there do appear to be meaningful differences in the behaviors of mice whose cannabinoid receptors are being stimulated. So, the data suggests that there is something to the claims that there are meaningful differences between the sort of massive variety of strains of cannabis in terms of their effects, but it's far, far more complicated than just the ratios of THC to CBD. Okay. So this group did a whole bunch of pharmacological experiments to try and figure out if those terpene molecules interact with cannabinoid receptors on their own, right? Independently of any Win55, any cannabinoid that we know is going to stimulate cannabinoid receptors. They did some interesting pharmacological experiments to get a sense of that. Even in the absence, of any molecule, right? CBD, THC, right? I don't think it would be particularly edifying to go through those data just because they're really nitty-gritty pharmacological experiments, and we'd have to explain a whole lot of stuff that isn't necessarily particularly interesting. But the short story is that when they checked out if these terpene molecules might be able to interact with cannabinoid receptors, just like in a test tube, let's say, it looks like they might be able to on their own. So it's possible that these terpenes, the molecules that are the main reason that different strains of cannabis have different odors, it's possible that they not only influence the nature of effects that strains have due to them containing THC and CBD, but that they may even interact with the same system that the main active ingredients of cannabis influence, right? And so in other words, they may have their own meaningful cannabinergic effects, cannabis stimulating effects. And they may contribute, right, to the overall effect of cannabis by binding cannabinoid receptors on their own, or modifying the reaction of cannabinoid receptors when they're bound by more classic cannabinoids like THC and CBD. Okay, pretty cool. Okay, so another neat thing about this study, they explored whether these molecules interact with a certain receptor that you may never have heard of, but you're almost certainly intimately familiar with the role it plays in your daily life and are very likely antagonizing this neurotransmitter receptor at this very moment. If you've had any coffee, any tea, any Coca-Cola today, you're antagonizing this receptor, the adenosine receptor. Okay, so what do coffee, tea, and Coca-Cola have in common? They're all drugs, of course, or more accurately, they all have a drug in them, caffeine. The stimulant caffeine, one of the most widely consumed drugs in the world, happens to have a pretty strange pharmacological effect as far as stimulants go. It's an antagonist at an adenosine receptor. And so in other words, caffeine basically prevents that specific receptor, that specific adenosine receptor, from being stimulated. It basically like gets in the way and prevents, the neurotransmitter that would normally bind it, adenosine. It prevents adenosine from binding its receptor, okay? And that's what stimulates us. That's why caffeine is stimulating, because the neurotransmitter, adenosine, builds up as we're awake for longer and longer, sort of like a biological clock. The longer we're awake, the more adenosine is building up in our brain, binding adenosine receptors and making us sleepier and sleepier, okay? But with caffeine on board, it's like our brain has no clue that this neurotransmitter is building up because caffeine's preventing it from binding. Okay, so that's a sort of adenosine receptor in a nutshell, a very, very small nutshell. It's all way more complicated than that because it acts as a sort of counterbalance to dopamine and regulating body movements, which is why it's actually a target for the treatment of Parkinson's disease, believe it or not. But the point is that this lesser-known receptor is associated with how much energy we will feel like we have, as well as how much we'll tend to move, as well as the quality of our movements, okay? So for example, when someone's just had 2 cups of coffee, right, or an espresso or a Red Bull, energy drink or whatever, you might see their knee bumping up and down, right, just jittering. And that's because there's a whole bunch of dopamine and other catecholamines flowing, okay? But basically 0 adenosine receptor binding because there's just a ton of caffeine standing in the way, okay? So, okay, I know that this isn't necessarily a particularly sexy story, but hopefully that gives you a sense of, why the data this group collected regarding the terpenes and the adenosine receptor are intriguing. Okay, so let's get to it. So I know that this is a super duper uncool figure because it doesn't get much more boring than just bar graphs, but I understand, but just stick with me. This is actually pretty cool. So what we're looking at is how much their mice moved around. Okay, it might sound like a pretty ridiculous thing to measure, but when it comes to mice, they tend to move around, walk around, at about similar levels, okay? It's a little bit more complicated than that, but they tend to just behave kind of similarly under similar conditions. They tend to have similar baseline movement, behaviors. So we're looking at how much time they moved around. The higher the bar, the more they moved around. And you see that black dotted line at the top there, right there? That's how much the mice normally boogie, right? That's how much they normally move around. And so that's the sort of like baseline mouse behavior. Okay, and then to be clear, like every one of these treatments, each of the bars, the orange bars, is slowing the mice down, but some way more than others. So let's start with the bar that's sort of stripy, with the star on top, right? Ischidaphalene is how you pronounce that word underneath that bar. So Ischidaphalene is a Parkinson's medication that has been used actually for a while internationally, but fairly recently approved in the United States. So it's Parkinson's medication, and I know that sounds like a whole can of worms, but you can really think of it as basically being like caffeine, but like a way more targeted version of caffeine at a specific receptor. So it's sort of like the difference between throwing a dart at a dartboard and throwing a basketball at a dartboard. Ischidaphalene is the dart, caffeine is the basketball. Okay, so ischidaphalene will tend to prevent hypolocomotion because it blocks things from binding that adenosine receptor, kind of like caffeine, right? It just gets in the way. And so, if something is trying to bind that receptor, but ischidaphalene is around, no dice, no binding. It can't bind the adenosine receptor. And as you can see, that bar is pretty high compared to the other ones, right? Okay, so now on the other side of the figure, we're seeing geraniol, right? One of the terpenes found in cannabis, the one that smells sort of rosy and is present in citronella. That's, just straight up geraniol alone, right? Nothing else. The mice were given nothing else. So now do you see that red dotted line? Okay, the sort of lower one. That's the effect of Win55, that cannabinoid that might as well just be THC. And I know that there's a ton of this drug might as well be another drug going on, but there are some good reasons for why they went with these very specific molecules rather than the more familiar caffeine and THC. But anyways, so you see that red dotted line, and you see how much Win55 causes mice to just sort of relax and stop moving around so much. Okay, that's Win55, the effect of Win55. Geraniol alone caused them to relax even more than Win55. Right? That's what we're seeing with that solid bar. It's a little bit lower than the Win55 red dotted line, right? But then right next to geraniol, that checkered bar, That's what happened when mice got both Geraniol and Win55, a combination of the two. And as you can see, they tended to get even more chilled out, right? Now, the white bar is pretty informative, okay? And here's where I would have done things a little bit differently in this figure, but rimonabant is essentially the opposite of THC or Win55, okay? It's like if THC or Win55, is stepping on the brakes, rimonabant is the gas pedal, right? And as you can see, that there's really no big difference. It's like it doesn't really matter that rimonabant is on board when mice are given Geraniol. But then we return to that Ischidaphalene stripey bar, right? All of a sudden, major change. The mice aren't nearly as chilled out. And in fact, they're statistically significantly less chilled out, right? They're moving around way more, almost as much as just a sober, normal mouse. So what does this tell us? Why are we doing all these combinations? Or why did this group do all these combinations? It suggests, first of all, Geraniol seems to have some interesting effects on its own. Also, there seems to be an interesting interaction with Win55, which means that there seems to be an interesting interaction between Geraniol with cannabinoid receptors, or at least cannabinoid receptor activation. However, it also seems that rather than exerting its chillaxing effects via cannabinoid receptors, Geraniol seems to at least depend upon adenosine receptors, the ones that Ischidaphalene block, depends on adenosine receptors being available and not blocked. Okay, and we can tell from that stripy bar, right? That's the only treatment that prevented this reduction in locomotion. Okay, so there's a ton more to this study. Okay, they did this kind of experiment where with basically like all of the tetrad, cannabinoid effects for each of the terpenes that they tested, right? They did a thing like that, but instead of hypolocomotion, it would be analgesia, or it would be whatever, hypothermia. And what they found is that they seem to be capable, these terpenes seem to be capable of inducing cannabinoid-like effects on their own, and that some of the terpenes seem to have some kind of interesting interaction with the adenosine signaling system. The story's more complicated than that, as it always is, but I'll be honest, this was pretty surprising to me that these terpenes would actually have a meaningful effect on behavior. So there are a few provocative things about this study. Okay, first of all, that whole notion that these smelly, sticky terpenes actually have some meaningful influence on the effects of cannabis and cannabinoids, and therefore, cannabis itself, that actually contradicts results from past studies. So they have some pretty reasonable explanations for why it is that they think they came to these conclusions, these opposing conclusions, while past studies came to the opposite conclusions, which in part revolve around their explanations revolve around the high concentrations of the terpenes that were examined, as well as how they treated each terpene individually, right? As opposed to just combining them all in a soup and just sort of cranking up the concentration of just one in that soup, right? Instead of like honing in on specific terpene at a time. Okay, so that's part of why it's provocative. It contradicts past literature. But it's also provocative because their data suggests that these smelly terpenes seem to add to the effects of cannabinoids, right? They have effects on their own, which is provocative, but then when they're combined with cannabinoids, there can be an additive effect. Pretty interesting. And then, of course, as always, there are things about the study that are worthy of criticism. That's always the case. And I do think, they might be pushing their interpretations, frankly, a bit further than I would, just based on the data that they're showing. But it's still a pretty interesting set of experiments. And, I'd love to see a broader set of terpenes examined, along with some more, molecular and circuit research. But so what's the answer to the question? Does the smell matter? Does the smell of cannabis matter? Does the scent of cannabis affect the effects of cannabis? And the answer to that question, at least according to these data, seems to be yes, which honestly is pretty cool. And I'll admit that my assumption has always been, well, of course it matters, right? But only insofar as the odor of anything influences the experience of that thing, right? But it's not like there's a meaningful pharmacological difference between what happens in my body when I eat a Honeycrisp versus a Red Delicious apple, which I'd never ever do, to be clear. But these data suggest that there's not only a sensory experience of, one strain that's unique to that strain, smelling unique, right? Rather, the same thing that causes that unique odor seems to be associated with unique pharmacological effect that would therefore be unique to that strain. And given, that one of the cannabis tetrad effects happens to be analgesia, or the reduction of pain, this possibility that terpenes can at least influence the efficacy of cannabinoids themselves may help to, the efforts to more scrupulously and accurately identify optimal strains of cannabis for folks who are looking for ways to manage pain, with a medication that isn't an opioid. And of course, that's just one possible application, right? So pretty exciting stuff, very interesting, totally surprising to me, I'll admit. But yeah, I hope you enjoyed, and that's what I have for you today. So if you dig this kind of conversation, maybe you'll consider subscribing and liking and all that kind of stuff. Otherwise, thanks for watching, and I will see you next week. Oh, Scott, hey, thanks for hopping in, man. Are terpenes present in hops? Yes, hops too. Lots of IPAs are described as dank. Yeah, exactly, right. I got one called Benjamin Danklin. And another called Sticky Icky Icky. Yeah, there's a ton. A bunch of the Philly breweries have names for like that too. And yes, absolutely. And in fact, one of the terpenes that they studied, alpha-cumulene, in cannabis, right, it smells like hops. And of course, the hops plant is pretty similar to the cannabis plant. So yeah, pretty cool. All right. Thanks for watching, everybody. Maybe you'll consider subscribing if you dig it. Otherwise, I will see you next week. See you, everybody.